Good morning, everyone. Welcome back again. Yeah. So I hope you have enjoyed the sessions we had for last 2 days, especially with me. And obviously, the series is going on where in which you can study and learn a lot more important things, and hence you can boost up your preparation for the NEET exam. So with me also, we started the topics 2 days back. If you remember, last to last class, we have completed the topic of Galileo's formula related to motion in a straight line, motion under gravity. In our yesterday's lecture, we talked about the dynamics of circular motion, in which we had a discussion of what types of acceleration they come into the picture whenever you talk about the circular motion. How does their magnitude vary? How does their direction vary? On what factors they are dependent? And our second phase of learning of dynamics of circular motion will be centripetal and centrifugal forces that we will study in our regular classes. Okay, so that was just the introductory part. Similarly, today I'm going to take a very important topic of fluids. Fluid dynamics or fluid mechanics is one of the chapters which the NEET aspirants tend to leave by the end of the preparation. Not because the chapter is very tough. The questions which come from these chapters of fluids and surface tension related properties, these questions are basically very easy that come in the final exam. But yes, since the topic is a bit different, you can't feel it every time, and that's why there is a tendency of majority of students to either not prepare the topic up to the desired level or majorly to leave that topic. So today we are going to fight with our phobia or fight with our tendency, so that fight with our nature of leaving the topic, so that at the end of the preparation we can perform in these questions also. Number one. Number two, the main problem with these fluid topics are that you know them, you know the fluids, you deal with the fluids in everyday life, but still you can't feel the mechanics involved. Okay, so today let us try to whatever we study in theoretically, let us try to feel the topics and let us try to relate it with our experiential learning, what we experience in our everyday life. Let us try to relate both the things. So physics से भाई feel आनी चाहिए, तो we will work on the similar grounds. Okay, so let us start here. So today I have taken the topic of surface tension, but I will tell you something regarding the fluids chapter first, like how to prepare it for the NEET exam. If we talk about the fluids, I generally classify the fluids into three parts. One will be hydrostatic. So when we teach the fluid topics here, we divide them into these categories: hydrodynamics, and then we talk about surface tension and related properties. Okay, this thing will be a very important checklist for you for your NEET exam preparation, so that you can have a clear-cut idea in which category a certain law, certain principle like Pascal's law, Bernoulli's theorem, equation of continuity, they fall, and how to prepare them. What are the fundamentals required to study them? Okay, so hydrostatics mein we will talk about the hydrostatics mein we are going to talk about ideal fluid at rest. Okay, now the fluid should not move with respect to the container, but the container itself can entirely move. I mean, you have kept some ideal liquid in a jar, in a beaker, and now you are accelerating the beaker or moving the beaker with a uniform velocity in a certain direction. All those things will be considered under hydrostatics. The fluid should not be moving. Okay, but the container in which the fluid is kept, in normal language, you can say, yes, that can move. Hydrodynamics. Ideal fluid or ideal liquid, particularly we will talk about here, in motion. Ideal fluid ही ले लेते हैं. Since I have used the name hydrodynamics, so hydro term is generally related to water too. So ideal liquid in motion. Ideal liquid in motion, ideal fluid or liquid. Okay, in motion. And here surface tension and related properties, this is applicable for real liquids. This is applicable for real liquids. Real liquid. Now, what is the difference between ideal liquid and real liquid? You need to understand here. Ideal behavior is every time easy to understand. Okay, easy behavior, not a complex behavior. But real things have some complexity involved. So it's every time easy to understand and deal with the ideal things. Consequently, then you can talk about the after understanding the difference between real and ideal, then you can also talk about the real things. So first of all, I'm going to deal with properties of ideal liquid. Properties of ideal liquid. So there are 4 properties of ideal liquid. First is ideal liquid is incompressible. You cannot compress an ideal liquid. So if you cannot compress the ideal liquid, its density cannot be changed. I mean, if you compress a liquid, definitely the particles will come closer and hence the density, the degree of closeness will increase. If you expand it, its density can be decreased. But if it is incompressible, its density can't be changed. That is, you will say density rho is going to be constant. Density is going to be constant. It won't change. Number 2, non-viscous. Non-viscous. So there is no viscosity. But sir, in fluids we study the viscosity. Yes, we do study the viscosity, but viscosity is applicable only for real liquids. Day-to-day life we deal with the liquids. Those are all real liquids. Okay. So non-viscous means the coefficient of viscosity is 0. No surface tension. No surface tension. Surface tension is also not there. But sir, you have kept the heading of surface tension. That's what I am saying, that surface tension and related properties, excess pressure, viscosity, everything will come under the category of real liquids. They are not applicable for, these properties are not valid for ideal liquid. So no surface tension means surface tension T or S. S or T, we denote the surface tension with either S or T, that is going to be 0. Number 4, irrotational. The angular velocity of liquid molecules is not there. But anyways, we are not going to deal with this property in detail for NEET syllabus. As of now, we can understand that yes, overall, these are the 4 properties we are getting for the ideal liquid. For real liquids, obviously these will not be, these properties will be available. Like viscosity you will find, that is non-viscous. Here we will find the viscosity, here we will find the surface tension, here in reality we can compress the liquid. So we are dealing with the compressible liquids. Okay. I hope that's okay. Right. So this is a clear-cut idea. If I talk about the topics that come under this category, hydrostatics mein you talk about the Pascal's law, you talk about the Archimedes principle, you talk about the hydrostatic pressure. Okay. Hydrodynamics mein you talk about the conservation of mass, which is manifested, which is expressed in terms of equation of continuity. You can talk about conservation of energy in a moving liquid, which is expressed as Bernoulli's theorem. Then the application of Bernoulli's theorem, Magnus effect, Venturi meter, blood pressure, blowing of the roofs, dynamic uplift, moving of aeroplane, Torricelli's theorem, speed of efflux, aspirator pump, many things will come into the picture. But today our major concern is surface tension and related properties. Surface tension and related properties mein we will talk about the excess pressure, viscosity, many things over there. So as of now I am talking about surface tension particularly. Let me, I would suggest you to take a screenshot of this so that you will remember how to categorize the topic of fluids. So if you are good with the categorization now, you have a, you know how to study, then studying this topic is going to be very easy here. Okay. And every year the question, in fact this year also the question came in the NEET exam from the surface tension topic only, and I'll take up that problem also. As of now you take the screenshot here. Okay. Chaliye, let me take you forward. Now today we are talking about surface tension particularly. So what do you mean by this term surface tension? The term itself is trying to give you a hint. What does it mean? Surface tension means a kind of tension created on the free surface of a liquid. Free surface of a liquid. So I will say property of a liquid means we are strictly talking about real liquid now. Property of a liquid by virtue of which its free surface. Free surface means that surface which is exposed to the air. Its free surface behaves as a stretched membrane. Basically a tension is created on the surface as a stretched membrane. And how does it, why it behaves as a stretched membrane? To minimize its surface area. And why there is a need to minimize the surface area, we will talk about that. As a stretched membrane to minimize its surface area. This property of a liquid by virtue of which its free surface, property of a real liquid, every liquid you can say, in day-to-day life every liquid you see is a real liquid only, because of which its free surface will always behave as a stretched membrane, so that there will be a tension created on the surface. And what is the need for it? We will talk about the cause of surface tension. We will talk about it. The cause of surface tension. Now, basically what you need to understand is, here we will also talk about the intermolecular forces. So the way you study the intermolecular forces in chemistry, you have the forces like dipole-dipole, dipole-induced dipole, ion-induced dipole. Then there are London forces, dispersion forces, or van der Waals forces, right? So there are 4-5 different categories of the forces which you study in order to understand your interaction in chemistry. But I won't say that chemistry molecules are different and physics molecules are different. No, that doesn't make sense, right? So molecules are molecules only, sir. So as far as physics is concerned, we categorize those intermolecular forces into 2 categories. One is cohesive force, one is adhesive force, and you have heard about it because you talk about these forces, I believe, in biology too. So intermolecular forces, you categorize them into 2, cohesive and adhesive. Cohesive forces are forces, those are among the same type of molecules. Among same type of molecules. And if you talk about adhesive forces, that is among different type of molecules. Different types of molecules. So why we are talking about here intermolecular forces, sir? Because the root cause of surface tension is cohesive force only, these cohesive forces. Okay, so let us see how it is happening. And before we proceed further from here, you need to understand that any intermolecular force here, F intermolecular, the way your gravitational force in physics and Coulombic force shows inverse square law, they are generally showing a rapid degradation. It decreases, it depends on R to the power 8, intermolecular separation to the power 8. Means that is, if your intermolecular separation gets doubled, if your separation gets doubled, then force magnitude of force reduces by or reduces to how many values here? 2 to the power 8. 2 to the power 8 means 2 into 2, 4. I'll just mention it that way. It's a huge number. It's a rapid degradation. Reduces to 1 by 2 to the power 8 times. 1 by 2 to the power 8 times. I hope you can understand this. So basically what I'm trying to tell you is that eventually if you separate two charges, you know that the magnitude decreases rapidly. If you separate two masses, you know that there is a decrement in magnitude, huge decrement. It's a rapid degradation. But if you particularly talk about the intermolecular forces, the degradation is far much rapid, right? R to the power 8 it's a inverse proportionality mein, it's a huge value. Getting the idea? Are you getting the feel that if the molecules are even separated a bit more apart, the intermolecular interaction between them will degrade very, very rapidly. Okay. And here the cohesive forces, cohesive category of intermolecular forces is somehow serving as the reason for surface tension, cause of surface tension. Let's see how it is coming as a reason. I will say that this is a beaker in which there is a real liquid filled, and I'm going to draw some layers. This is suppose the free surface of the liquid. This is the free surface of the liquid. So it is exposed to the air. Okay. Being any molecule, if I am a liquid molecule, then I can interact with my neighboring molecule on this side, 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 every side, right? All the directions I'm going to I'm going to get attracted towards the molecules, and I'm going to attract the molecules from all the directions. Understood? Now, but there is a particular range that beyond this range, I'm not going to interact with further molecules. I have a range, that up to this extent only I can attract and I can get attracted. So that range for a molecule is called range of influence, R, range of influence R. So I'm going to save some space here. And just a moment. Yeah, so here R is the range of influence, and that will serves as radius for sphere of influence. Now what does that mean, sir? I said being a molecule, I'm going to attract or put some attractive forces in all the directions. Okay. Now if being a molecule, if I'm going to put some attractive forces in all the directions, this is going to give you a spherical symmetry. A sphere it is available because of symmetry. So if the distance up to which I can put my attractive effects or I can get attracted, that will be called as a radius of influence. So this spherical symmetry will be known as the sphere of influence. Okay. So that serves as a radius for sphere of influence. So I will say, let us suppose that this is the range of influence R, and this is another layer. Okay. Now consider few particles here inside. Suppose I consider a surface molecule. Then I will say a surface molecule M or P. A surface molecule P is experiencing only and only attraction towards the bulk. A surface molecule P is experiencing only and only attraction towards the bulk. A molecule which is at a distance like range of influence Q, so it will experience some attraction. Up. Let us consider another molecule first, somewhere here. I'll change the color so that it will be clearly visible. Q. So can I say there is a bit of attraction in the upward sense, but there is a dominating attraction still in the downward sense. Okay. And now I will say, suppose I consider another molecule R here, another molecule capital R here, exactly at a distance of range of influence, then you can feel that it is symmetrically attracted in upward sense as well as downward sense. So there is no net attraction force experienced by it. And if you again draw some S molecule here, again it will experience net upward and net downward force exactly identical, exactly of the same magnitude. So what I'm trying to show you is that a surface molecule will only experience a net downward force or a net force towards the bulk, I should say appropriately. A molecule in between this region, you can see some small arrows directed in the upward direction, but bigger arrows directed in the downward direction, which clearly shows that there is some attractive force experience in upward direction also, but there is a majority of downward attraction. So P will only experience the force towards the bulk. Q will dominating force will be experienced towards the bulk, but for molecules R and S, the net force experience is going to be 0, because the force experience down must be balanced by the force experience upwards, or I can say force experience towards the bulk, but the bulk is available in all the directions. The bulk is available in all the directions, and hence you can say there is no net force experienced by the molecules R and S. There is some net force experienced by the molecule Q towards the bulk, but there is a huge net force experienced by the molecule P towards the bulk. As a result, we can conclude that all the surface molecules are going to experience a huge force towards the bulk. All the surface molecules are going to experience a huge force towards the bulk. If all the surface molecules experience force towards the bulk, they all want to go in the bulk. So no one is ready, no one is happy at the surface. No surface molecule is happy at the surface. No molecule is ready to stay at the surface. Everyone wants to get inside the bulk because of this net attractive cohesive force. Cohesive hi to hai, that is cohesive only because it is liquid molecule is attracted towards another neighboring liquid molecules. So it is among the same type of molecules. Are you getting it? So what we have concluded is that ultimately, because of the cohesive force, the net force experienced by surface molecule is towards the bulk, and that's what the cause of surface tension is. If no one wants to stay at the surface, everyone wants to get inside the bulk, then surface will always have a tendency to minimize the surface area. Because of the minimization of the number of molecules who wants to stay there, you can say you want to minimize the surface area. Okay, and hence there is a tension created on the surface. That tension is called the surface tension. I hope the idea is clear. So this is basically the surface tension in case of real liquids. Surface tension in case of real liquids. I hope you are good and clear with the concept of surface tension. Now, I'm going to express something else too. So basically, this surface tension is a property. If we imagine, if we consider an imaginary line on the free surface, we will talk about it. We'll talk about this property on the next slide, where we will study surface tension as a physical quantity. Here we have studied surface tension as a property. We have studied surface tension as a property. So I will attach another tip here. For a given volume, sphere has a minimum surface area. Sphere has a minimum surface area. And hence all the droplets, all the liquid droplets tend to attain spherical shapes. For example, I will say that suppose I am considering a small drop and then I am considering a big drop. So I will say in case of a small droplets, definitely you will have perfectly spherical structure. Okay. In case of big drops, you can say because of the gravity, the middle portion can be a bit flattened, but the curvature will still be observed at the ends, like that. Okay. So basically, you can clearly see what is the meaning of surface tension as a property. What is the root cause of the surface tension? The cohesive type of intermolecular forces. what are the categories of intermolecular forces and how does it vary, how does it show the variation with the distance. And obviously we will talk about that for a given volume, why we always talk about spheres, spherical droplets, dew droplets are also, dewdrops are generally sphere. Mercury when falls on the floor, suppose you have experienced that a thermometer has been broken in front of you, the mercury thermometer, maybe in your childhood or maybe the recent experience, you can feel that whenever it drops, it spreads into the, it breaks down into the droplets. Spherical droplets only, why always the drops are majorly observed to be spherical in shape. The reason for that is also in front of you. I hope the idea is a bit clear. Okay, so you just take the screenshot for it, then we will proceed with the other aspect of surface tension, how the surface tension can vary. I mean, what will happen if the surface tension will be more? What do you want? If you want to wash the clothes, you want the surface tension to be high or surface tension to be low? If you want the liquid to flow, you want the surface tension to be high or surface tension to be low? How the surface tension varies with temperature, all those things varies with impurities, detergents, soap, these things we are going to study. So just take a screenshot, then we will talk further. Okay, I hope you have taken the screenshot. So till now we have understood how to classify the fluids, what are the properties of ideal fluids, ideal liquid, and then we have talked about the surface tension as a property. And now let us talk about we will also talk today surface tension as a physical quantity. Surface tension as a physical quantity. As a property we have studied, as a physical quantity we will also study that, then we will make use of these things again and again. So before this, I will tell you the factors affecting surface tension. Factors affecting surface tension, because we have already got the idea of surface tension as a property. So now let us see what are the factors that affect this property of surface tension. Number one, we will talk about the temperature. We will talk about the temperature, theta, because T say to we are going to, by T we are going to denote the surface tension. Number two, we are going to talk about the impurities. Now in chemistry, you would have studied that whenever you talk about the impurities, there are always two categories of impurities: sparingly soluble impurities and highly soluble impurities. Sparingly soluble impurities and highly soluble impurities. So let us categorize them here. Category A, I will say sparingly soluble, sparingly or partially soluble impurities, and then we will talk about highly soluble impurities. Highly soluble impurities. Okay, and similarly, like let us talk about them one by one. First of all, I would like to talk about the temperature. I will say if temperature is increased, if temperature theta increases, then intermolecular separation R increases. If temperature will increase, they will move randomly, the intermolecular separation will increase, and hence this implies force, intermolecular force decreases, cohesive force decreases. This implies surface tension will also decrease. Okay, so if you increase the temperature, then surface tension decreases. A mathematical relation can be written as surface tension final will be equal to surface tension initial 1 minus alpha theta final minus theta initial. Okay, surface tension final will be surface tension initial 1 minus theta final minus theta initial. Here this minus sign is denoting that there is an inverse relation. I repeat, I am not talking about inversely proportional. I will say inverse relation inverse relation between temperature theta and surface tension T or ST or S maybe. Okay. Once again, what I'm trying to say is that if you increase the temperature, surface tension will decrease. If you decrease the temperature, surface tension will increase. And here alpha is named as temperature coefficient. Alpha is named as temperature coefficient of surface tension. Temperature coefficient of surface tension. That's the name of alpha. Okay, that's the name of alpha. Temperature coefficient of surface tension. Okay, so there is an inverse relation. What are the common examples, sir? How we can feel it? Again, I am telling you, the topic of fluid will be stronger only if you try to feel it. Whatever you are writing, whatever you are studying, try to relate it with the common examples. For example, example number 1, I would say, example number 1, hot food tastes better. Hot food tastes better in comparison to cold food. In comparison to cold one. The reason is, if you have cold food, cooler, lesser temperature, surface tension will be high. So if you eat cold food, if you have cold soup or cold liquids, you won't be able to taste it in a good way. The reason is, because of the low temperature, because of the high surface tension, it will be coagulating together. It will minimize its surface area. It will not spread on your taste buds over the tongue. Okay, a bit of biology. Okay. So basically, if you talk about hot food, temperature is high. Temperature is high, surface tension will be less. Surface tension will be less, it can easily spread. High surface tension, coagulation tendency, minimizing surface area. Low surface tension, spreading easily, tendency. So if it's a low surface tension, it will spread on your taste buds easily. And if it spreads on your taste buds easily, you are going to enjoy the taste. You will be happy with the taste. Okay. Number 2, if we talk about washing the clothes. Now, if you want to wash the clothes, you want the clothes should easily wet. The clothes should be easily wet by the water. So water should not be having a very high surface tension, because if it is having a high surface tension, it won't spread over the clothes. It won't wet the clothes easily. It will try to minimize the surface area. That's not good for washing the clothes. So it's easy to wash the clothes. It's easy to wash the clothes with hot water. As its surface tension is low, and hence high wettability. Okay, I hope you understood the idea. Okay, if you haven't heard about this term wettability, now onwards you should understand. It is the wetting ability of a liquid, how much it can wet the solid. Or here we are particularly talking about the ability of water to wet the clothes, dirty clothes. Okay, so it's easy to wash the clothes with hot water, because with cold water, cooling, low temperature, high surface tension. Cold water will have high surface tension, it won't spread easily. Hot water will have low surface tension, it will spread easily, wet the clothes easily, and hence washing is easy. I hope you got the idea. So this is how you can remember, you can relate it with your day-to-day life experience, that how the surface tension varies with temperature. Okay, and similar type of experiences you will get when you when you try to dissolve some sort of impurity. What is that exactly? Suppose I say sparingly soluble impurities, partially soluble impurities. For example, camphor or soap detergent in water. Let's talk a bit of chemistry. Water is a polar protic solvent here. And soap and detergent are long chain fatty acids. So they are non-polar. Majorly, there is not a huge polarity. Long chain for fatty acids, so they are not highly soluble in water. Okay, so they are considered as a sparingly soluble impurities. And if you talk about the highly soluble impurities, then you will say example NaCl. NaCl, sodium chloride, common salt in water. NaCl and H2O, highly soluble impurities. So if you introduce sparingly soluble impurities, here surface tension decreases. Okay, and highly soluble impurities, here surface tension increases. That's why if surface tension will increase, salts will be there, surface tension will increase, wetting ability will reduce, and it's not easy to wash the clothes. Okay, it's not easy to wash the clothes, but if you put some soap and detergent, then it is very easy to wash the clothes. So surface tension decreases. So example 1, you will say that it's easy to wash the clothes. Again, here I am talking about washing the clothes with respect to temperature, and here I am talking about the washing of clothes or wettability of a liquid in terms of the dissolved impurity, and that dissolved impurity is sparingly soluble impurities, soap and detergent. So it's easy to wash the clothes with water containing soap or detergent. Okay, one example. Another example, it's a previous year question. I'm giving it to you as an example here. So if you take an irregular shaped camphor over the surface of water, particularly why water I am taking, because I told you that camphor in water is always is also a sparingly soluble or partially soluble impurity. So if you consider that here there is an irregular shaped camphor, why there is a need of taking irregular shaped only? Because a regular shape might assure that dissolution is uniform and symmetric from all the sides. But if it is an irregular shape, then definitely there will be an irregular dissolution. If there is an irregular dissolution, sometimes the dissolution from the left hand side will be more. So if the dissolution of camphor on the left hand side will be more, the surface tension from the left hand side will reduce. Again, I'm repeating it, it's a sparingly soluble, it will result in decrement in surface tension. So if from the left hand side the surface tension will reduce, so the camphor will fall like this. On this side, if the dissolution is more, the surface tension will reduce, the camphor will fall like this. So this irregular shaped camphor will keep on dancing, because of the decrement in the value of surface tension locally. Now I just use the term locally. Generally you will hear about the two terms locally and globally. Globally refers to a change which has been brought to the entire sample. Global means complete. Local means particularly in that area, that region, or in its neighborhood. Okay. So here we will talk about the dissolution of irregular shaped camphor results in the decrement of the value of surface tension locally. As a result, till the time it is not completely dissolved, it will keep on dancing like this. Random movement. I am giving it the name of dancing. It is just referring to the random movement which will be observed here. I hope the idea is clear. And what will happen if you consider the highly soluble impurities like NaCl in water? You will say surface tension value will increase. If surface tension will increase, coagulation tendency will increase, spreading tendency or wettability of the liquid will decrease. Hence, it's difficult to wash the clothes with hard water. Okay, so I'm taking the example of washing the clothes again and again because that is a very common example which you experience in day-to-day life. All right. I hope the idea is clear. You can quickly take the screenshot. Then we'll move ahead. Okay. So I'm right now telling you to take the screenshot so that you can revise the things in between your breaks also. But remember, there's a Telegram link given in the description of the video which you can click and join our Telegram group. Okay. So this is surface tension as a property. What we have studied today, first of all, a quick revision here. First of all, we have understood that fluid topic has to be divided carefully in the category of ideal liquid behavior and real liquid behavior. These are the properties mentioned for ideal liquid. So we will talk about hydrostatics, where ideal liquid will be considered to be at rest. The container itself in the liquid is there, can easily move. Okay. Then hydrodynamics, and but here today's target discussion is surface tension and related properties. Okay. Then we have understood the surface tension as a property. What do you mean by that? Where it is used, what is the root cause, and how you can practically experience it. All the dimensions we have covered. Then we have understood that what are the factors which are affecting the surface tension. Okay. So our next target topic will be surface tension as a physical quantity. So I hope it's time almost to complete the discussion. So let us meet very soon, and then whenever we are taking the topic of surface tension once again, our target will be from this surface tension as a physical quantity. It's a very simple formula, and based on which this year NEET exam question also came, that there is a disk which you want to uplift from the surface of the liquid. How much is the additional force required due to surface tension? So it won't take much time, but right now it's time for other subject discussion. So let us hope that we will meet with the same discussion once again in near future, and we will definitely discuss that. Okay. So till then, enjoy your break. All the best. Bye.
Good morning, one and all. So, in our previous lecture, we have started with the chapter Morphology of Flowering Plants. There we talk about the organ of the plant. What is that? Means angiosperms, that is the root. We discussed that, right? Now, please put your thumbs up to show that whether the voice is audible. And just this word written, the stem, today's topic is clear to you or not? Just put your thumbs up. Okay, it means it is clear. If you are not responding, I am taking obviously it is clear. So let's start with the topic, that is the stem for today. But before that, a small recapitulation from our topic, the root, what we have discussed, right? The journal characters we have taken, or the root, there I said you it is underground, it is non-green. We are taking its journal characters, right? So exceptions are there. We are not taking that right now in these two character what I spoke. So here it is positively geotropic and negatively phototropic. I given you the word tropic and trophic. Tropic is the growth plus movement. Trophic, PHIC, which is coming at last, that means related to food. But here we are talking about tropic, TROPIC, means movement plus growth, right? So that we discussed. Even we discussed that like stem they don't have, means I must say unlike the stem, they are having the features different from them. Like in the roots they are means nodes and internodes are absent. In roots the nodes and internodes are absent. Even buds are absent. One exception I have given you from the bud. Can anyone respond me here please, that which example I have quoted you for the root where the buds are going to be present? Exception. Epomia is an example for that. Please do listen the lecture and make a habit, especially for the repeaters I am talking about. Keep in habit of listening the things again and again. This is the only tool how you can maintain your botany till your NEET exam, right? If you are not reading, revising and listening the things, maybe near to the NEET exam, all the learned thing is going to be very much dull, right? So keep on revising, keep on listening the things, right? So, okay. So we discussed about the root, then we discussed about the types of roots. Their types of root we discussed: taproot, we discussed about fibrous root, adventitious root. Taproot mostly present in the dicots and fibrous in case of monocots. Adventitious root, I quoted you the example of banyan tree and even for monstera, right? Then taproot is bearing the primary root with the various branches and various degrees of the other orders of branches they are having. Whereas in case of the fibrous root, initially the primary root develops from the radical part, but later it will disappear and gradually, slowly, a tuft, a bunch of the roots are going to create at the base of the stem. In adventitious root, the origin is other than the radical. That all we have discussed. Then regions we discussed. Base part is towards the ground and the tip part which is more penetrated in the soil. Do remember that thing we have discussed. And if I go from tip to the base of the root, that means first is the region of meristematic activity, region of elongation, then region of maturation. That all features we have discussed. Then we have discussed about modification in roots. There we discussed the taproot modification and the adventitious root modification. In taproot modification, the function based on that we take storage, respiration and for the nitrogen fixation, right? Then for the adventitious root, we have taken the functions storage, extra support. Under extra support, we discussed about the prop roots and the stilt root, right? So that all points we had discussed. Now we are going to start our today's topic, that is the stem, which is our today's topic, means next organ in the flowering plant. Now what is there? in the stem, let's talk about its journal character and compare it with the root. This is next important thing. If I am taking the journal characters of a stem, you must know that its origin is the plumule, and origin of the root is what? Radical, right? And next thing, it is the ascending part of the axis of the plant. Main axis of the plant, it is ascending, means it is moving towards the light. Its growth is happening towards upper side. It is a ascending part of the main axis of the plant. And moreover, the stem, it is positively phototropic. Again, let me to say you, photo means the light, right? Keep on analyzing the thing here. Here the photo means light. Tropic means growth plus movement. Both is happening. Here the word is not trophic. Even I am repeating this again and again. They will make a trap of the question based on that words only, right? Now next after this, it will be negatively geotropic. Right? Against the gravity it is going, or away from the ground level it is going. Then after that, you must know they bears what thing over there? Nodes and internodes, right? And can I say they even, let me to write here, presence of even one more thing, nodes, internodes, as well as the buds. Right? In my yesterday's session, I said you what is the difference between nodes and internodes. Now today we will take buds, what are these and which type of bud will be present. Let me to clear this thing first of all. If I am taking this part here for the buds, I am taking the next slide for this, for the buds. To understand the buds, you must know one point, that in the stem, we are having the node part. The node part is that where the development of leaves happen. Where the development of leaves happen. This region, this is what? Node part, right? And between the node, what is present? What is this thing now? This will be internode, right? Now what is this? Buds. This is a leftover meristematic cell which is present between the angle of this stem and the leaf. Between this stem and the leaf part in this angle. What is present over there? Axillary bud. Right? Or even you can call it as lateral bud. Right? And what is this thing now? Where it is present? At the node part. Right? So buds are going to be meristematic, and here the axillary bud, what I have shown you here, it can give rise to the flower even, right? Or even it can give rise to the branch, right? If it is giving rise to flower, you call it as floral bud. Otherwise, you call it as vegetative bud or the stem bud, right? And which is present at the tip, part, that you call it as apical bud, or you call it as terminal bud, right? So this is the feature. Now if I show it through one diagram over there, then naturally how it is present, you will find like this. Can you see it here? It is what? Terminal bud. See here, this is a terminal bud. This is what? Lateral bud. What is the other term I have used for the lateral bud? Axillary bud, right? So you will find the presence of bud as a main feature of the stem. But in roots, that buds are not present. Exception in the roots I have given you for the epomia, one of the examples in the exception I have said you. So here this is the presence of bud, and these are the node part, this is the leaf, angle of the stem and the leaf, there is a presence of the lateral bud, right? 1 point. Now bud can be adventitious. Yesterday I said you the word adventitious means what thing? The origin is other than what is present. That is the adventitious. Like we said yesterday, adventitious root means origin is other than radical. Now bud origin is what? That is the, no doubt, apical meristem that you will learn in the chapter anatomy of the flowering plant. But just now it is developing further at the angle of this aerial part, like this, at the angle of this stem and the petiole part here, or at the tip of this. But now it is on the leaf. As an example I have shown you bryophyllum, leaves are having now bud. The place is not the stem, the place is now on the leaves. That's why you are using here the word adventitious, right? So which type of buds these are? Adventitious bud, because it is not in that angle or the tip, it is on the leaves now. It is not only on the leaves, it can be, like I said you, as an exception in the roots, like epomia roots they are having, even that will be adventitious bud. But which is present on the leaf, like I have shown you here in the diagram, on the leaf, that you call it as foliar bud. And which is present on the roots exceptionally, that you call it as the what type of bud? That is the root bud, right? That type of bud is adventitious again, which is not in the angle, which is not on the stem now, it is other than the stem. Here on the leaf, that's why you call it as the foliar bud, right? And exceptionally it can be on the roots, as per I said you. Now this is the feature of the stem what we have discussed. Let's talk about further more features. This feature just now I explained, which is one of the journal character for the stem. Now coming to the next one, when you see the stem in the young stage, it will be green. It will be photosynthetic green. Whereas if I am talking about dicot stem, you know that it will show secondary growth and it will become brown and woody in case of the dicots. So can I say that cases the young stem will be green, but older one will become brown and woody. In the plants where it will not become brown and woody, it remain green, you call it as herbaceous stem, which means it will be having the green color and photosynthetic, right? So these things you should know about the journal character. One more interesting thing you should know here, lateral branches, their lateral branches are exogenous in origin, right? Means it is formed from the cortex. If you cut the transverse section of a stem or of root, you will find the various layers of the cell. Innermost of the steel part is the pith, means central is the pith, outermost is the epidermis, right? Between the cortex and the central part, there is a pericycle, right? I am talking about the steel part. Steel part is consisting from the pericycle till the pith, right? Let me to show you here. I am talking about this epidermis, outermost layer. When you cut a stem, you will find outside epidermis. Then inner the epidermis, what is there? Here is a cortex region. And this you call it as pericycle, right? So whatever is there now in the pericycle, that part you call it as a steel part. Are you getting that? So in the anatomy we will discuss the points that in the steel you will find the pericycle, vascular bundles, medullary rays in case of the stem, or conjunctive tissue in case of root, and central part is the pith over there, depending upon which section we are talking about. But generalized thing is that and means whatever is coming including pericycle, that you call it as the steel part. Now why I am saying that point? Reason, if anything is originating from any part of the steel, that will be endogenous in origin. Anything which is originating from outside this steel part, that you call it as exogenous in origin. If I said you the lateral branches are exogenous in origin, that means it should not be from pericycle or any of the cell which is just inside to the pericycle. It should not be from there, right? So from where it originates? Either from epidermis or from cortex. Now it is originating from the cortex. Are you getting that? That's why you call it as exogenous in origin. But in roots, branches are coming out from pericycle. That's why in the root, the lateral branches are endogenous in origin. This point is somewhere is linked to the anatomy part, right? If you are not able to carry this point right now, just wait for the anatomy portion, just wait to come over here, we will help you out in that thing, right? So just now, for a moment, journal characters of the stem, to remember their lateral branches are exogenous in origin, whereas in roots, their lateral branches are endogenous in origin in case of roots. Okay, so these are all about the journal characters of the stem. Now, next after this, if I want to show you the functions of the stem, what can be the function of the stem? Number one, the production of the branches bearing the leaves, flowers and fruits. They are giving branches so that the main parts of the plant, that is leaves which is for photosynthetic, flowers is for the reproduction and further forming fruit and seeds, that is an important thing. To bear that, they are having the stem. Next, it even helps in conduction of water, mineral and photosynthase. What do you mean by the term photosynthase? Means the products formed after the photosynthesis, that you call it as photosynthase, right? So photosynthase, the major one is what thing? You know that sugars. For the conduction of that, even stem helps, right? And then storage of the food can be there in case of the stem. Then after that, it helps to support, to protect for the vegetative, and even for they are for vegetative propagation, right? So three function, but this is slightly for the modification process. I am going to discuss after that the modification. First 2 points right now given on the board are the major points of the primary function of the stem. Major these two. That is present, that are the major function, primary function of the stem. In the last point, what is mentioned, this is mainly for their modifications. We will discuss over there, right? Okay. Now in this, all that points of the stem, we just started talking about their modifications. Yesterday even I said you, what do you mean by modification? When an organ changes its structure and their form, obviously they will not perform primary function. They will try to give rise to the other functions, that you call it as secondary functions. Like in root, we have discussed for the storage, we discussed nitrogen fixation, we discussed about the respiration, we discussed about the extra support, and many more are there. Now here let's see that what other function they can perform other than these primary function of the stem, right? Let's check on that. So for that thing, you must know about a flowchart first of all. Their modification is divided under the three headings. The first heading, that is underground stem modification. Then after that is the subaerial stem modification. Then after that, aerial stem modification. These three headings are there. And see, this topic have been deleted from your NCERT book, but it's somewhere very again important, here and there asked in the different chapters in one way or the another way. Or next thing very important, in your practical books, they are still mentioning some of the topics of your NCERT which have been deleted. Still we have to carry forward that thing, right? When you will come here, definitely we will guide you that which thing you have to read from your old book and which you should not at all, right? This topic slightly somewhere links here and there, so you must know about these things. We cannot teach only one part out from the main topic, but you should know all the headings, only then you can find the difference, right? So on that aspect, to cover all the questions types in your NEET exam, we have to touch some of the deleted part as well, right? So be careful, this is important. Then after that, underground, what modifications are there? Under the underground modifications, we will take the rhizome, we will talk about the bulbs, we will talk about the tubers, we will talk about the combs, right? Under the subaerial, we will discuss about the runners, we will talk about the stolons, we will talk about the suckers and offsets. And here in aerial, we will take the concept of the phylloclade, cladode, thorns, right? See, I am not writing spines here because spines are the modification of leaves. Here, these are the thorns, to remember this thing. Then even we will discuss about the tendrils, and even we will discuss about the valvules, right? So these are the various underground stem modification, these are subaerial stem modification, and these are the aerial stem modifications, right? Now to go with detail with all these modification, basically first of all what you should know, what do you mean by underground stem modification, subaerial and aerial? This part first of all you should know. Now what do you mean by underground stem modification? Suppose this is an ground level. If any modification is below the ground, obviously that will be underground stem modification, right? And suppose this is a ground, if any modification is touching to the ground, near to the ground, that is going to be which type of modification? Subaerial modification. If I am taking this as a ground level, and here if I am taking the modification, like suppose this is a stem part, now it is away from the plant, like suppose for example thorns, it is away from the ground level, it is not touching to the ground like this. This you call it as a aerial stem modification. In subaerial part, some part can grow down even, again it can come up, but it is near to the ground only, touching to the ground, it will be like that. That you call it as a subaerial, which is completely under the ground, obviously it is underground stem modification, and aerial will be, that will be obviously not touching to the ground, on the stem away from the ground level, that is aerial stem modification. Fine. So that is the basic difference between underground stem modification, subaerial and aerial stem modification, right? So just to clear that point in your mind, I should say you the difference between these three headings first of all. That I said, now we are going to take the underground stem modification, where we will discuss all that things. Now for that I am taking help of this diagram. There first of all, let's talk about the rhizome. What is an example shown over there? That is of ginger, right? So here the rhizome is an underground stem modification. Rhizome is an underground stem modification. Now what is the role of this rhizome? That helps in the storage of the food. No doubt this is even for storage, combs are even for storage, bulbs, stem is not for storage. Their leaves are for storage but not the stem, keep it in your mind, right? So here even the tubers, their stem are even for the storage, right? So this is the common thing for the storage function for rhizome corm, and for tubers. Bulbs I will take later. So I am taking first of all for this storage. Then how this rhizome is different from the corm and from the tubers, right? From the corm, how it is different? The corm can grow slightly vertical down the ground. The rhizome are growing more in the horizontal position. Are you getting that? So here it is more growing in the horizontal position under the ground. Now why you call it as a stem modification? Because you will find on that the nodes and the internodes. And on these nodes there is a presence of the scaly leaves. Scaly leaves will be there. And from the nodes there is the origin of this adventitious root. Even they are having adventitious root origin from there. On which point? On the node part, right? See the difference in the question, what they can keep. For example, already I said you, these three are for the storage, but its stem is not for storage. Now, what is the difference between rhizome and corm? The rhizome can be more growing in a horizontal fashion, and this will be growing more under the ground vertically. It is growing more in the vertical position, right? This can be long, but this can be short in its length, right? And here nodes and internodes, here nodes as well as internodes which are framing, obviously they are present, but you will find the node part are more concentric. It will be more concentric one after the another. Here it can be irregular. When you will see the ginger or even the turmeric, you will find their nodes are not concentric. They can be like this. They are not properly concentric, right? So these are the major differences between this rhizome and the corm, right? Much more are even there, but right now I am talking about the main superficial thing, how you can identify this is the rhizome and this is a corm. This is short and having a little bit more reserve food material and growing vertically down. It is growing vertically, it is not vertically, it is horizontally, and it is more longer in length as compared to the corms, right? Now potatoes, even it is for the storage, right? But here on the potatoes you will find nodes, right? On the nodes further, what is there? There is a presence of the buds. On the nodes, there is a presence of the buds. Now this node plus bud, that you call it as eye, right? What do you call it as? Eyes. I repeat this point once again. What is an eye on a potato? Eye consists of the node part plus axillary bud. Axillary bud, that you call it as an eye. Getting that? So in the case of tubers, you will find these eyes are there and there will be no adventitious root coming out from here. There is no adventitious root coming out from the tubers part. Adventitious root you will find in the rhizome. Even you will find adventitious roots in case of corms. Even you find adventitious roots in case of bulbs, right? But in the tubers, you will not find adventitious roots. Are you getting that? See, let me to say this point once again. The rhizome, corm, bulbs, they will show adventitious root. But storage of food will be in rhizome, corm and tubers. Are you getting that? This is very important fact that you should know. you should know for the underground stem modifications, right? So this is the basic differences between all these points, that these rhizome, they are having the nodes, internodes, no doubt, because it is a stem modification, but it is not concentric, it is irregular. But here it will be concentric. Both are having adventitious root, but in tubers there is no adventitious root. Adventitious roots are even in case of the bulbs. Getting that? Now coming to the bulb part. In the case of bulb, the storage is happening in case of leaves. Then what is about the stem? Stem is a reduced and discoid. Discoid means it is like a disc. Nodes and internodes will be like this. And from the each node, leaves are coming up. And you know that leaves are for the storage. Leaves are for storage. These are the nodes, right? So concentric type of the nodes will be there in case of the onion in their bulb, means in their discoid stem. Whereas the proper you are having the stem like structure in the case of rhizome, corms and the tubers. But here it is going to be reduced and become like a disc in case of the bulb, right? So this is the underground stem modifications that you should know in case of the stem. Let's recapitulate this point. Underground stem modification. First we discussed rhizome. Second, we have compared it with the corms. Then we compare it with the third one, that is the we taken like a corm. Then I repeat for the rhizome, and even we have taken from the tubers, right? So that three we discussed. Why? Because all three are for the storage. All three will not have adventitious root, because in tubers adventitious roots are absent. And corms, even the rhizome, they have adventitious root. And even in the case of the bulbs, you will find the adventitious root, not but in case of the bulb, in case of the tubers, you will not find adventitious root, right? So this is the important thing that you should know. Now coming to the next modification, that is the subaerial stem modification. Subaerial stem modification, I just said you it will remain close to the ground part. Let's see the first one. They have given you for the runner, right? Runner, let me to clarify first thing, it is happening in case of the grasses, right? In grasses, what is there? The stem part is running along with the ground level, and this elongated thin part, that you call it as a runner. This part you call it as runner. And on its node part, this is a node now. From the nodes, what is coming up? Leaves. And what is down to that? The tuft of the roots will be coming out. It will be running throughout the ground. And due to intercalary meristem, the grazed part again can be bring back again. It will start establishing in the new niches. So can I say the subaerial modification are more for vegetative propagation, right? More for the vegetative propagation, right? Just now I am telling you about the effects of that structure. I will come back on all the topics once again with their examples as well. Right now I am clarifying the terms for you, that what are the runners, what are the stolones, right? All the points we are discussing. Now just I put the example, reason why, just to start with the thing and you can easily compare it with each other, right? So runners are running on the ground and spreading in a new niche. Niche means the new region adjoining to that the grasses keep on growing. On the node part, upper side will be the leaves, downside it is having tuft of the roots, right? Now coming to the next thing, right? Where is that? Stolones. Now stolones are what? Now please listen this point very carefully. In stolones, what is happening? Running along the ground, the stem part, when node is there, no doubt leaves will be there and the roots will be down. But the branch, one of their branches will start moving above the ground like this, but it arches downward, means it bends downward, and where it touch to the ground, again it give rise to new plant and give rise to the root of the new plant towards the ground. If it bend and form the arch, if arch is going to be formed, right, if arch is going to be formed, due to that the bending position of this stem, that you call it as a stolon part. Getting that? Now what are the suckers? These two things are always confusing. Stolons are running above and bending down, touching new plant, right? Whereas suckers, running part of the stem, one of the branch start moving obliquely upside, means of the parent plant body. This is suppose parent for this plant. It give rise to the obliquely going upside the branch. What is that? Obliquely placed branch, right? When it is start obliquely playing, it will not arch down, it will keep on growing vertically up now, right? So once it keep on growing, when it obliquely comes out, that you call it as what? Suckers. But once it bend down, that you call it as stolon. And what is coming up after coming obliquely from down part, that you call it as suckers, right? All these three modifications we have discussed in case of the terrestrial plants which are on the ground. But in the case of aquatic, you are having the modification which you call it as offset. Offset now is almost similar like runners. That is running on the ground. This is running on the surface of water. But it will be short. This offset will be short and thick. And the node part, you will find rosette of leaves. What do you mean by rosette of leaves? Means leaves are arranged like the rose petals. That the rosette of leaves will be appear on the upper side, and downside you are having the roots, which is giving buoyancy, being these are the aquatic plants, that upward thrust will be given by their roots, right? So these four modifications I have said you for subaerial stem modification. There first we have learned about runners, then stolons, then suckers, then we have discussed about the offsets, right? Now let me to say for the example for subaerial, then I will go back for the underground stem modification. I will complete that topic with the examples, right? So here one example I said you for grasses. Next example you should know about the oxalis. For the stolons, the examples will be the mint and the jasmine. I am taking example from your NCERT book only. Suckers, I am writing here. For the suckers, example, chrysanthemum will be there, even banana, even the pineapple, right? And here offsets, example of offset, Iconia and even the pistia are the examples for this, right? Pistia and Iconia are the example of offsets, right? So must know the basic concept on this topic. The next level is even their example. I am completing the underground as well by giving you the examples on this. I am writing in this flow chart itself for the underground. For rhizome, what example just now I quote you? Turmeric and even the ginger. For the bulbs, onion and even the garlic. In the tubers, you have learned the example of potato. Which tubers I am talking here? Obviously the stem tubers. Corms, example will be colocasia, right? And even Amorphophallus, right? So underground stem modification I have written here. and subaerial I already spoke. What should be the example over there, right? Now last topic of the day, from the stem we are going to take their modification. What do you call it as? Aerial stem modifications. Aerial, I said you, that modification which are not touching the ground, it is not under the ground, that you call it as the aerial stem modifications. Let's take the first, let's take the meaning of all, then we will go with the examples. Phylloclades. Keep in your mind the word phyllo stand for the leaves. Clade is for the stem here. When the stem is becoming leaf-like, not only the main stem but even their branches will become leaf-like. They will have the chlorophyll and it will show the photosynthesis, such type of stem modification that you call it as phylloclades, right? Now let me to compare it with the cladode. Can you see in this diagram of phylloclade, the main stem and branches both are photosynthetic, whereas in cladode, the main stem will remain as such, but only branches. Now see, these are not leaves, these are the branches which is becoming photosynthetic, right? What it has become? It has become photosynthetic. When it branches become photosynthetic, only branches, main stem remain as such, that modification you call it as cladode. Phylloclade, when stem and the branches both become photosynthetic. One word is more there, phylloid. Phylloid is a leaf modification. Under the leaf modification, we will discuss that point, right? Just now we are discussing the term phylloclade, right? This is what thing? Phylloclade, right? Even here I mark, and here we are discussing about the cladode. Cladode only branches, phylloclade, main stem and branches both will be there, right? Now coming to the next modification, that are the thorns. These are the axillary bud modification, right, which is for the protection. Now their function is the protection here. For these two cases, the function will be photosynthesis. That's why it is the modification here, right? And it is helping in protection. Thorns. Keep in your mind, thorns are the stem modification, spines are the leaf modification. Both thorns and spine are deep-seated. Deep-seated means they have the connection with the vascular bundles. One word is there, prickles, right? Prickles are not the stem modification and even it is not a leaf modification. It is just an elevation of the surface of the plant. For example, in rose you will find prickles, but in case of the bougainvillea you will find the thorns, right? And if you go with the cactus, it will have the spines. Thorns and spines, they are deep seated, means they are connected with the vascular bundle, right? Just now I have discussed with you the phylloclade and cladode and the thorns. Now third one I am going to take for the stem tendril. It is functioning for the support. Which support? To which type of stem it is giving support? Which is weak. That stem need the support, and again the axillary bud will give rise to this tendril. This is again axillary bud modification, right? So this function will be support here. Now coming to the last modification of aerial stem modification, that will be bulbil. Bulbil is what? Bulbil is helping in the storage, right? And wherever this axillary bud modification, this is again the axillary bud modification, wherever it is going to fall down, it gives rise to new plant, right? That's why it helps in the vegetative propagation and making the plant to persist even in the adverse condition, being storage of food is there, which is feeding even the new plant when it is about to grow from this bulbil, right? So these all are the aerial stem modification we have discussed. Now let's talk about their examples. Now for the examples, again I am using this flow chart. Right here I am going to write phylloclade. What example? That will be the cactus. Do remember in Opuntia, which is one of the example under that, it is having flattened type of the phylloclade. Whereas in case of Euphorbia, it is having cylindrical, right? And let me to show you through the diagram once again. By what thing it is protected? It is protected with the help of spines. And do remember, leaf spines obviously is the leaf modification. So spines, you should not take it under the stem modification. It is for the leaf modification, right? So do remember that point. And here, now these things are for arid type of plant. Arid means which are living in the dry conditions. That's why the leaves are going to be having less surface area, being modified into the spines. Then who is performing work of photosynthesis? Stem, which you call it as phylloclade. So these example I have written for the phylloclade, right? Now for the cladode, what will be the example here? The cladode example will be the ruscus and even the asparagus. And thorns example bougainvillea and even the citrus. For the tendrils example, you must take for the like watermelon, which comes under the gourds. Even pumpkin is there, that watermelon comes over there, that comes under the gourd. And even the grapevine, even it comes under tendrils. Bulbils example will be agave. Agave will be the example, right? So these are the example for what thing? This is for the aerial stem modification, right? Subaerial, again let me to complete this flow chart. Okay, let me to write here subaerial. What example for runners? That will be the grasses, even the oxalis. Stolons example will be mint and the jasmine. Suckers example will be chrysanthemum, banana and even the jasmine. What are the example for offsets? That will be including the eichhornia, which you even call it as water hyacinth. Even you call it as terror of Bengal, right? Water hyacinth. Even you call it as terror of Bengal. Even next example will be what thing? Pistia. So all that example twice I have written and discussed with you. I hope so you get it now. So 3 modifications we have discussed today for the stem under the heading underground stem modification, then subaerial stem modification, then the aerial stem modification, right? So in this way we have completed our the topic of the stem, and do compare with the root and come out with the things in comparison and even individual what is there in that organ. Today we have taken the next vegetative part of the flowering plant, that is a stem. Previously we have discussed about the root. In our next lecture we will start for the leaves, right? This is all for today. Thanks a lot, students.
క్లాసెస్లో Yes. Good morning to all. So we are going to continue that yesterday's one only. So next topic is here, types of neurons. So there are different types of neurons are there in organisms. The first one is unipolar, next bipolar, pseudo-unipolar, multipolar, and one more is there, that is known as apolar neuron. So like that there are different types of neurons are there. And this classification, it is based on number of dendrites. This classification is based on number of dendrites and axon. First one is here, unipolar. In this unipolar, it has only axon. It has axon but no dendrites. So it is known as unipolar. In the case of bipolar, it has dendrites. It has dendrite as well as it has axon. So for that reason, it is known as bipolar. Next, pseudo-unipolar. In this pseudo-unipolar also, there is no dendrites. It has axon, but axon is branched in nature. Because of branched nature, it is known as pseudo-unipolar. It looks a little bit different here. And multipolar. Multipolar has many dendrites. It has many dendrites and has one axon only. So this is a different types here. So where do we find this unipolar one? Means unipolar, it is present in embryonic stage. This is important NEET question. Unipolar neuron, it is present in embryonic stage. Next, bipolar. Bipolar neuron, it is present in retina of an eye. It is present on retina of an eye. Next, multipolar. Where do this multipolar present? Means multipolar is present in cerebral cortex. Cerebral cortex. In NCERT, we have unipolar, bipolar and multipolar is there. And extra one is this pseudo-unipolar. This pseudo-unipolar is sensory in function and it is present in dorsal root ganglia. And it is present in dorsal root ganglia of spinal cord. And next one is here apolar. Last one is apolar. In the case of apolar, here we cannot differentiate the dendrite and axon. This is just like a primitive cell, primitive neuron. This primitive neuron we can observe in lower organisms like hydra. It is present in hydra as well as we can observe in our eye also, that is known as amacrine cells of eye here. So this is the classification of different types of neurons. Next one here. What is PNS? PNS means peripheral nervous system. This peripheral nervous system consists of nerves. It contains all nerves arised from CNS. Here, PNS means here it consists of all nerves which are derived from or which are arised from CNS. Okay, there are two types of nerves are there. What are those two types of nerves means here? Nerves are of two types. One is afferent nerves and second one is here efferent nerve. What is this afferent nerve means? The afferent nerve is also known as sensory. It is also known as sensory nerve. And whereas afferent is known as motor nerve. It is known as motor nerve because it has motor in action. So what is the role of this afferent nerves means? This afferent nerve, it transmits impulses. NCRT line, this one, important one. It transmits impulses from tissues or organs to CNS because it has a sensory role. Sensory role means here it will transmit the impulses from sensory organs like eye, ear, nose, etc. directly to the CNS. So it is known as sensory nerve or afferent nerve. Second one is efferent. Efferent is motor in function. Motor in function means even this one also transmits impulses from, this is written means it is from CNS to concerned peripheral tissues or organs. Yes. Are you understanding, children? Harshini? So what is afferent nerve means here? It transmits the impulses from tissue or organs to CNS. But whereas efferent nerve means it transmits impulses from CNS to concerned peripheral tissues to organs here. So these two are the types here, afferent and efferent. Here. So can you identify what is the first one here? First one, here it is carrying from tissues or organs to CNS. So this is known as afferent. This is afferent pathway. This is afferent pathway. And the second one is here from CNS to, this is CNS. From CNS to concerned peripheral tissues or organs. This pathway is efferent. Efferent pathway here. Next one here. Can you identify this picture? In this picture, it is showing afferent as well as efferent here. So whichever impulses are coming from body to CNS means afferent, and from CNS to body means here that is efferent here. Okay, and next topic is we are going to discuss about central nervous system. Central nervous system means here it constitutes yesterday classification we have done. It constitutes brain as well as spinal cord. And what is the role of this central nervous system? This central nervous system which acts as NCRT line, it acts as central information processing organ. Central nervous system has brain and spinal cord. Out of these two, what do you call the brain means? The brain is a central information processing organ and it acts as command and control system. Important points, these two. So this is the brain. So what is the role of brain here? The brain plays an important role here. Number one, it involves in number one, voluntary movements, And number 2, even it will regulates the involuntary organs. Even it regulates the involuntary organs. And next, even it plays an important role in thermoregulation. Next, hunger, thirst. Next one is here, even they can regulate the endocrine glands also. Endocrine glands. And next one, circadian rhythm. Circadian rhythm means here that is known as 24-hour cycle. That is day and night cycle here. The brain plays an important role in voluntary movements and regulation of involuntary organs, thermoregulation, hunger, thirst, and next one is endocrine gland function as well as circadian rhythm, etc. Here. Not only this, even it will involves in, it plays an important role in hearing. Next one is sensing, vision, emotions, next memory, etc. Communication, thoughts, etc. here. So this is the role of brain here. So for that reason, the brain is known as central information processing organ. Next, here the brain is protected by. The brain is protected by, important point. It is protected by skull. It is protected by skull. Next, inside the skull, if you cut the skull here, this is skull. If you cut the skull, inside the skull, the brain is covered by. Protected by means skull is the answer, and it is covered by means answer is, it is covered by how many layers means 3 layers. 3 layers. These 3 layers are called protected by 3 layers. Those 3 layers are known as cranial meninges. Cranial meninges here. So the brain is protected by skull and it is covered by, where are these cranial meninges are present means they are present inside the skull. It is covered by 3 layers called cranial meninges. What are those 3 layers means here? This sequence important. The first one is dura mater. Second one is arachnoid matter, because it looks like a spider web. That's what arachnoid matter. The third one is here pia matter. So where is this dura matter is present means here, it is present outer side. So outermost layer is dura, this one. This is dura matter. Outer layer is dura matter, and next one is here middle layer, and actually it is thin. It is a very thin middle layer known as arachnoid matter, and third one, innermost one is here pia matter. This pia matter is highly vascularized and it is very close to the brain tissue. Okay, these are the 3 meninges. Order is from outer to inner means order is dura matter, arachnoid and pia matter. Suppose when the question is from inner to outer means reverse we have to write here. Next. This is the brain. The brain has divided into 3 major regions. It is divided into, the brain is divided into 3 major regions. What are those 3 major regions means here? Number 1, forebrain. Next one is midbrain, and last one is here hindbrain here. Next one is here forebrain. What are the points comes under this forebrain means here? The forebrain is, it is a major part of the brain. It means it is the largest portion of the brain. Major part or larger portion of the brain is here forebrain. And this forebrain has structures called, first one is cerebrum. Second one is here thalamus. And the third one is here hypothalamus. This is also NCERT point. Brain has divided into three major regions. Out of that, the forebrain is the major part or is the largest portion. And this forebrain has a structures called cerebrum, thalamus as well as hypothalamus here. And out of these three, the cerebrum is the largest one. Out of three, forebrain is the major one. And out of these three under forebrain, the cerebrum will come. This is the large sized, or it is a large portion or major portion of the brain here. So this cerebrum, number one, cerebrum here. Cerebrum is the major one. The cerebrum has divided into two equal halves. It is divided into two equal halves. Those halves we are calling as cerebral hemisphere. It is known as cerebral hemisphere. It means right and left cerebral hemisphere. Suppose cerebrum is there like this. So to this cerebrum, in middle a deep cleft will be there. A deep cleft which cuts or which divides the cerebrum longitudinally into two equal halves. Those two equal halves is known as cerebral hemispheres. And these two cerebral hemispheres, they are connected to each other. They are connected. These two are connected to each other by means of tracts of nerves called, very, very important point this one, this is known as corpus callosum. It means two cerebral hemispheres like right and left cerebral hemispheres joined together by means of a corpus callosum to form into a one whole structure. That whole structure we are calling as cerebrum. So this cerebrum is the major portion of the brain or even major portion of the forebrain also, correct. Next, under cerebrum only, a layer of cells. A layer of cells covers the cerebrum. A layer of cells covers the cerebrum called, it is called cerebral cortex. So what is cerebral cortex means? Cerebral cortex is nothing but a layer of cells which cover the cerebrum is known as cerebral cortex. So how do this cerebral cortex appear means? This cerebral cortex, it is present in the form of foldings. In the form of foldings. It is present in the form of a foldings here. For example, in this previous picture, this one. So all these are the foldings. So these foldings we are calling as outer layer of cerebrum. It is known as cerebral cortex here. Cerebral cortex. Next, the outer region of the brain is grey in appearance. It is known as grey matter. The outer layer is known as grey matter, and whereas inner side this is known as white matter. It is white matter. What is the reason of this grey color and white color means here? This grey color is mainly due to presence of because of concentrated cells. So this is assertion and reason question. So why do the outer region of the brain is called gray matter? The answer is here because it appears grayish in color. Why it is in grayish in color means because in this region there is more number of cell bodies, or they will have a concentrated cell bodies, and which appears that grayish color. What about inside? Inside the color is white. So why it is white in color means here because inside it has myelinated tracts. It has myelinated tracts here, and this myelin sheath which appears opaque and white in color, so that the inner regions of the brain is white in color and it is known as white matter, but outer layer is grayish in color and it is known as gray matter. Here even this is also same. So this is outer region is gray in color and this is inner one is known as white matter. And inside the brain here it has some cavities. These cavities are known as ventricles. It is known as ventricles. This is the cross section of brain. Next. So this is one half means it is one cerebral hemisphere. It is one cerebral hemisphere. So the two cerebral hemispheres, they are joined to each other. They are joined to each other by means of a tract of nerve fibers known as corpus callosum. This is corpus callosum. Here they are showing. So this is the corpus callosum. This corpus callosum will join the two cerebral hemispheres into cerebrum. Next, what is the role of cerebral one? This is important point. Cerebral cortex has three regions. What are those three regions means? One is it has sensory area, and the second one is here it has motor area, and the third one is here it has associative areas. Here, motor area, sensory area as well as associated areas. The role of sensory area is it receives sensory impulses, and the role of this motor area is it involves in the transmission of motor or regulatory impulses, and associated area is here, this point is important. So associated area is it is neither sensory or motor. It is neither sensory or motor. Then what is the role of this one? It involves in some other activities. What are those means here? It involves in intersensory, and next one, even it will involves in memory and communication. And communication here. So this is about cerebral cortex. The cerebral cortex has sensory, motor and associated area, but this associated area is a large region. This is actually a large region, and this region is known as associated area, or it is neither sensory nor motor, and it involves in intersensory function as well as memory and communication. Next. It has a limbic system. Even this point also important. What is limbic system means here? This limbic system consists of, or it is made up of, number one, deep it consists of here inner regions of forebrain. So this part comes under here. So it contains inner regions of forebrain, and second one is here amygdala, and third one is here hippocampus. So what is this amygdala means here? This amygdala is known as emotion center. It is known as emotion center, where it will controls all our emotions. Inner parts of forebrain, and second one is here amygdala, emotion center, and third one is here hippocampus. Hippocampus is known as memory center here, because it recollects our memory. Here. So the limbic system, which consists of the inner parts of forebrain, amygdala, as well as hippocampus. Next, along with these three, hypothalamus. What is hypothalamus? Hypothalamus is known as master gland here. So along with these, hypothalamus involves in here. So these three as well as hypothalamus, they plays an important role. It involves in number one, regulation of sexual behavior, Second one is here regulation of emotions, emotion control, and third one is motivation. So what is limbic system means? Limbic system, it consisting of forebrain, amygdala, and hippocampus. Here all these three along with hypothalamus involves in regulation of sexual behavior, emotion, as well as motivation here. So these are all about cerebrum here. Next. So this is the picture. So this outer region we are calling as cerebral cortex, and this is the forebrain region. In this one here, the hippocampus is there, and then amygdala, emotion center, as well as hypothalamus, and some parts of forebrain also involves, and it is known as limbic system. Next, going back here, next one is thalamus. What is thalamus means here? A structure which is wrapped by cerebrum. A structure which is wrapped by a cerebrum is known as thalamus. And what is the role of this thalamus means? Very, very important point. This thalamus acts as a major coordinating center. It acts as a major coordinating center for sensory and motor impulses here. So which of the following structure of a brain acts as a major coordinating center for sensory and motor means? The answer is thalamus here. And last one is here hypothalamus. What is hypothalamus means? Hypothalamus is a structure. It is present. Hypo means below. It is present at the base of. Thalamus. Base of thalamus. A structure which is present at the base of the thalamus is known as hypothalamus. And what is the role of this hypothalamus means here? Hypothalamus plays an important role in controlling of body temperature, that is known as thermoregulation. And the next one is urge for eating. Urge for eating and also drinking, because it maintains homeostasis here. So, which structure is present at the base of the thalamus means hypothalamus, and the role of hypothalamus means here it plays an important role in thermoregulation, urge for eating as well as drinking here. And this hypothalamus has some special cells here. For example, here I will write hypothalamus has neurosecretory cells. It has neurosecretory cells. What is the role of this neurosecretory cells in the hypothalamus? These cells, it secrete hormones. Those hormones we are calling as hypothalamic hormones. Hypothalamus region has some special neurons. Those special neurons are known as neurosecretory cells. Why do we are calling it as a special neurons means? Because they are involving in secretion of hormones called hypothalamic hormones. What are these hypothalamic hormones means here? For example, releasing hormones, and next one is here, for example, one example I will give, GnRH. What is GnRH means here? Gonadotropin releasing hormone. Second one is inhibiting hormone. Inhibiting hormones. Inhibiting hormones means, same example you can take, gonadotropin inhibiting hormone, like that. Next, third one more is oxytocin. Oxytocin, which acts on uterus and it facilitates the easy delivery, that is parturition, and even it acts on mammary glands here and involves in ejection of milk out. And fourth one is here vasopressin. Vasopressin is also known as antidiuretic hormone or ADH. Here. Deficiency of this vasopressin or ADH leads to diabetes insipidus here. So, all these hormones are secreted from neurosecretory cells present in hypothalamus. So, the hypothalamus has a dual role. One is it plays an important role in thermoregulation, urge for eating and drinking, and meanwhile it also plays an important role in secretion of some hormones. Out of these releasing and inhibiting hormones, they directly acts on pituitary gland. So, the releasing as well as inhibitory hormones, they are acting on pituitary gland here, and these two hormones are secreted into pituitary gland, where it will be stored in neurohypophysis or posterior pituitary gland. So, this is about hypothalamus here. So, the forebrain portion is done. Next one is here midbrain. Next one is midbrain. So, what is this midbrain means? Midbrain is a portion, a small portion which is present in between. This is filling the blank question, important one. It is present in between thalamus or hypothalamus. Thalamus or hypothalamus of forebrain. So, midbrain means it is a portion and it should present between forebrain and hindbrain. So, it is present in which portion of the forebrain means either thalamus or hypothalamus, also correct, and pons of hindbrain. These two are important. So which structure is present in between the forebrain and hindbrain? Means the answer is midbrain. And next, this midbrain has a canal. It has a canal. That canal is called cerebral aqueduct. It is known as cerebral aqueduct. So where is cerebral aqueduct present? Means it is present in the midbrain and it allows the passage of cerebrospinal fluid. Next, third one. This is important point. Dorsal side of midbrain. So dorsal point important. Dorsal side of midbrain has some swelling structures here. How many are there? Means there 4 are there. It has 4 round swellings. Swellings, or it is also known as lobes. What do you call these 4 lobes? These 4 lobes we are calling as corpora quadrigemina. This is also important point. Corpora quadrigemina. Where is corpora quadrigemina present? Means it is present the dorsal side of the midbrain. And the role of this corpora quadrigemina is it plays an important role in visual reflexes as well as auditory reflexes here. So this is about midbrain. And next one is here hindbrain. Hindbrain has 3 portions. One is pons, cerebellum and medulla oblongata. Yes. So the last portion is hindbrain, and this hindbrain has 3 regions: pons, cerebellum as well as medulla here. What is pons means? This pons is a region where it interconnects. It interconnects the different parts of the brain. Which of the following structure interconnects the different parts of the brain? Means the answer is pons. Next, cerebellum. The cerebellum is called little cerebrum. It is known as little cerebrum, even if it has the outer foldings. Even it also has the outer foldings, that is known as outer folding structures called, that is known as cerebellar cortex. And even the cerebral cortex also appeared grayish in color. It also appeared grayish in color, and this cerebellum plays an important role in balancing of body. Here. So this is cerebellum, and last portion is here medulla. This medulla is the last portion, and this medulla continues as spinal cord. It continues as spinal cord, and the role of this medulla oblongata is it has some centers. What are those centers means here? It has a regulation on number 1, cardiac, next respiration, and even it involves in regulation of gastric secretions also. Cardiac activity, it involves in the regulation of cardiac activity, respiratory activity, as well as gastric secretions, here. So this is about hindbrain. Here, this is the picture. This portion is cerebellum. This is the cerebellum portion here, and this is pons, and this is medulla oblongata. This medulla oblongata, it is continuing as spinal cord. It continues as spinal cord here. So the cerebellum, which is present little bit outside will be present, and this is thalamus and hypothalamus. So these two comes under forebrain. Those two comes under forebrain, and this is cerebrum. This is cerebrum here. Next, brainstem. What is brainstem? Brainstem consists of, it has number one, midbrain. So the midbrain portion, next pons and medulla. So these three structures forms into a brainstem here. For example, in this picture, this is the midbrain portion here. In between, midbrain portion will be there. So the midbrain, next pons, as well as medulla. These three forms into a brainstem. So which portion of the brain doesn't comes under stem? This brainstem means answer is cerebellum. Cerebellum not involved. It is not involved. So which of the following structures are involving in the brainstem means only midbrain, pons, as well as medulla oblongata, but not cerebellum. And what is the role of this brainstem means? This brainstem, it connects. It actually acts as a connecting point. It connects brain and spinal cord. So a structure which can interconnect or which can connect the brain and spinal cord means the answer is here brainstem. So that's all for today. Thank you very much.
Right? Is it audible and visible to you? Good morning, everyone. Is it audible and visible to you? I hope it is audible. And today, yesterday, sir started how to make structure, right? So today we will continue the topic in a different manner, like how we can make structures, because structures will help you to find the hybridization, the shape, geometry, and many things, right? The main thing is how to make structures. If they gave you some compound ammonia, HClO4, so how we can make their structures, right? Many bond parameter questions and hybridization questions, all depend on structure. If you know, if you can draw, right? So let us start. First one is, our main part today is how to draw structure. Yesterday you have learnt one technique. I am discussing one more technique here. Fine. So first thing to draw the structure of any given compound is you should know what is the meaning of valence electrons, right? So first thing you should know what is the meaning of valence electron, right? Valence electrons is the number of electrons in outermost shell. Number of electrons in outermost shell, right? This information we should have, right? Like if I say in first group elements in alkali metals, valence electrons are 1. In second group elements, valence electrons are 2. In 13th group element, valence electrons are 3. In 14th 4. In 15th 5. In 16th 6. 17th 7. And 18th what? 8. So if you know your element belongs to which group, you can directly answer the valence electrons, right? Otherwise you can write the electronic configuration to find the valence electrons, okay? So like I am giving one example here, just one example I am giving for this. Imagine I am having here chlorine. Chlorine have overall 17 electrons, and if you write the configuration, it is 1s2, 2s2, 2p5. Now we have 3s2 and next is what? 3p5. So overall if we check, bache, the third shell is the outermost shell. The third shell is outermost shell, and it is having how many valence electrons? 7 valence electrons, because these 7, 2 these, 5 these are present in the outermost shell. Clear? Third shell is outermost shell, and you can say it is having 7 valence electrons. Or directly we can say, if our element like this chlorine belongs to 17th group, so if element is belonging to 17th group, you can directly say you are having 7 valence electrons. I am saying once again, first group elements have 1 valence electron, second group elements 2 valence electrons, 13th group element have 3 valence electrons, 14th 4, 15th 5, 16th 6, 17th group will have 7 valence electrons, and 18th will have 8 valence electrons, right? I will use this again in our topic. Fine. Now the next thing, the second point, if I take, these are the few elements. I will use 3 points only to draw the structures, okay? The 3 main points. The second point is, you know, second period. Second period, you can say it start from lithium, right? If I write the second period, I am writing here is a lithium. Then we can go for which one? Beryllium. Then we can go for boron, and like carbon, nitrogen, oxygen, fluorine, and neon, right? Out of these, most of the time you will see lithium though make very few or just LiCl type of covalent compound. Otherwise I will remove this also, and I will remove this also. So these are the second period elements. These are the second period elements which you will see in most of your compounds, okay? You should remember the second period elements do not have d orbital. So because of absence of d orbital, these elements, if these are present at center, as a center atom, so these elements cannot make more than 4 bonds. You can directly remember also. These few elements cannot make more than 4 bonds, right? Or you can remember element wise element, like which element? Beryllium, boron, carbon, nitrogen, oxygen. Or you can say second period elements will not make more than 4 bonds, right? So I will say the maximum covalency. The maximum covalency here, the maximum covalency of these elements is 4. These elements cannot make more than 4 bonds. So be careful about it. I am writing in one more way here. These elements cannot make more than 4 bonds. Okay. As a center atom, these elements cannot make more than 4 bonds. Why? What is the reason behind it? The reason behind it is, I will say, absence of d orbital, absence of vacant d orbital, or you can say absence of d orbital. Very, very important point. So we did 2 points up to now. To draw structure, we need these 2 points. First point, you should know what is the meaning of valence electrons and which element is having how many valence electrons. And the second point is, these few elements you should remember that these elements cannot make more than 4 bonds. Very, very important point. Clear? The next thing I am saying, the next thing is third point. I am writing some elements here which belongs to third period or below it, or most of the time in your test or in your module or NCERT you will see only few elements are repeating again. I am writing those elements. So like you can say the element can be silicon, phosphorus, sulfur, chlorine, bromine, iodine and xenon. These will be your center elements in most of your questions. Center elements will be these one or these one, that's it. In 99.9% questions, these are our center elements, right? So these center elements, these elements cannot make more than 4 bonds, we have to remember, but these elements can make many bonds, okay? So I am saying here, these elements can have covalency, more than 4. That means these can make more than 4 bonds, no issue. These can make 5 bonds, 6 bonds, sometimes 7, sometimes 8, no issue, right? I am repeating once, these elements will not make more than 4 and these elements can make more than 4. So these 3 points we have to be careful when we are drawing any structure, just 3 points, okay? The next one is, okay, I think all of you have done this part, right? Now, the next part is, I am writing here element, and here I am writing number of bonds that element form in general. Number of bonds in general. I am not saying here maximum, minimum, I am saying in general these elements make how many bonds. Fine? Clear? Just remember these few. Like if I am saying hydrogen element make how many bonds? 1 bond. In all of its compound you will see hydrogen always make 1 bond just to complete its duplet, right? If I come to oxygen in general make how many bonds? 2 bonds. Oxygen in general make how many bonds? 2 bonds. In general I am saying oxygen can make up to 4 bonds if it is center atom, but in general it make how many bonds? Only 2 bonds, right? If we come to 13th group, okay, or I will say here the second group element like beryllium, I am saying in general the beryllium make how many? I am circling the main part. It makes in general 2 bonds, but sometimes 3 possible, 4 possible, right? But you have to remember in general it make how many bonds? If I am coming to 13th group, it's like boron, aluminium, gallium, these elements mainly make 3 bonds. In general I am saying definitely more than that possible, right? And similarly, if you come to the next group beta, it is carbon, silicon type of things. So 14th group, instead of this I am writing group wise. So you can say 13th group elements normally make 3 bonds. 14th group elements normally make 4 bonds in general. 15th group elements in general want to form how many bonds? 3 bonds. 16th group elements in general want to make how many bonds? 2 bonds. 17th group elements in general want to make how many bonds? 1 bond. And 18th group definitely do not want to make any bond. An element can make how many maximum bonds? It depends, it belongs to which period. Like I gave already. If element belongs to second period, it can make maximum 4 bonds. If it belongs to third period or below it, it can make many bonds, no issue. And these are the in general. These elements make how many bonds in general in most of its compounds. This is that part. Okay. So if you remember these 3-4 points, it's enough for us to draw any of the structure, right? So I'm taking some examples, and we will try to see the method, the sequence in which we should follow, to draw all of our structure, right? So I am taking example here, very easy, okay? So first one I am taking here example. Imagine I am having here, you can say ammonia, NH3. This technique will help us to find the center atom is making how many bonds and how many lone pair it will have, which will help us to find the hybridization and remaining part, right? Tell me first of all, my first sequence, from where we have to start, the nitrogen belongs to 15th group and have 5 valence electrons. Nitrogen belongs to 15th group, remember that. And it have 5 valence electrons, fine. So as you can see, there is only 1 nitrogen and 3 hydrogen. So it's very much clear that nitrogen will be the center atom and hydrogen will be the surrounding. I'm saying once again, what is sequence? First of all, write the valence electrons of center atom. Now write the center atom and write the remaining atoms. Anywhere you can write, because here we are not discussing geometry or shape. Here we are discussing only how many bonds it will make or how many lone pairs it will have. So in most of the cases, remember this point, very important. In 99.9% cases, your surrounding atom will be hydrogen or oxygen or halogen. Hydrogen will make how many bonds? 1 bond. We know it very well. Hydrogen always make 1 bond. Halogens, chlorine, bromine, iodine as surrounding atom always make how many bonds? 1 bond. Oxygen make how many bonds? 2 bonds. So these 3 points are enough. Hydrogen make 1 bond, oxygen make 2 bonds, and halogens make 1 bond. That's it. So now carefully, as we know, the hydrogen will make 1 bond. So I am making 1 bond here, 1 here, 1 here. Out of 5 valence electrons of nitrogen, 3 are used in making bonds. So how many remaining? Only 2 remaining. Out of 5 valence electrons of nitrogen, 3 used in bonding, 2 remaining. These 2 become what? 1 lone pair. Okay, that's it. Very easy part. Clear? I'm saying once again, what is sequence? Write the valence electrons of center atom, then write the surrounding atoms, and now you know surrounding atom will always be hydrogen or halogen or oxygen, and we know hydrogen will always make how many bonds? 1 bond. So hydrogen making 1, 1, 1. In making 3 bonds, nitrogen used its 3 valence electrons, so 2 valence electrons remaining, that will come as lone pair. And remember, only the valence electrons can be used in making bonds, right? So out of 5 valence electrons, 3 are used in bonding and 2 become what? One of the lone pair, right? Clear? Now similarly, if I go for the next one, you see here, I am taking the PCl3. Imagine I am having PCl3. PCl3 is our next example. Tell me first of all, the main thing is valence electrons. So tell me, the phosphorus have how many valence electrons? 15th group element having 5 valence electrons. Phosphorus is having 5 valence electrons, right? So first of all, write the center atom. This is our phosphorus. This is our chlorine, chlorine, chlorine. You can write anywhere, in any surrounding, any side. Now I told you halogens as surrounding element always make how many bond? One bond. So chlorine is making how many bond here? One bond, one bond, one bond. We have to start from the first center atom, how many valence electron, then we look at the surrounding atom, and we know surrounding atom chlorine make always one bond. So 1 1 1 done. How many remaining out of 5 valence electrons? 3 are used in making bonds, 2 remaining. 2 valence electron remaining means it is what? Lone pair. Done? Clear? Clear, bacha? Next thing I will see, I can write here PCl5. I am writing PCl5. Yes, everyone? So if we are having PCl5, so here phosphorus is having how many valence electrons? 5 valence electrons. Again same thing. So first of all write the phosphorus here and write 5 chlorine surrounding it. You can write anywhere. Done? So we know chlorine to always make single bond. So you will see I am making here one bond, 1, 1, 1 and 1. In making 5 bonds, phosphorus used all of its valence electrons. So out of 5 valence electrons of phosphorus, nothing left. So I will say in this case, phosphorus will make only 5 covalent bonds and there is no lone pair on phosphorus. That's it. We should know the valence electron of center atom. Out of that, how many are used in bonds, and then remaining become what? Lone pair. Very, very easy, right? So I am taking some more examples like that. Fine. Next one. If we come to the SO3, assume I am taking SO3. Okay. Now tell me, sulfur is having how many valence electrons? Sulfur is having 6 valence electrons, right? Sulfur is having 6 valence electrons because it belongs to which one? 16th group. Now write the center atom and you can write your surrounding atoms nearby it. Tell me, yes everyone, tell me, oxygen make how many bonds in general? Means in 99% cases, oxygen make how many bonds? 2 bonds. Because it has 6 valence electrons, it want to make 2 bonds to complete the octet, right? So as I said, you should remember, hydrogen make 1 bond, halogens make 1 bond and oxygen make 2 bonds. That's it. So here I will say sulfur is making, sorry, oxygen making double bond here with sulfur, oxygen making double bond, oxygen making what? Double bond. That's it. That's it. Finish. Our structure is finished. Now careful. 6 valence electrons are present in sulfur and all these 6 valence electrons are used in making bonds. Will be there any lone pair on sulfur? No, because all 6 valence electrons of sulfur are used in making bonds with oxygen. So there is no lone pair presence. Clear? And one more thing we have to check carefully. Whenever you are making many bonds, you should see carefully. Your center atom belongs to which point? Second point or third point? I told beryllium, boron, carbon, nitrogen, oxygen, these elements, if these are present at center, these elements cannot make more than 4 bonds. But phosphorus, sulfur, silicon, or you can say the chlorine, bromine, iodine, xenon, these can make many bonds. Okay, so there is no issue. Sulfur though can make 6 bonds, no issue here, right? So it is completely fine. But nitrogen, carbon, these elements cannot make 6 bonds. They can make maximum how many? 4. So we have to be careful about it, right? Next thing I am saying, SO2. So please try SO2. Okay, this is SO2. And next, I am taking CO2. So please try SO2. Yes, try SO2 beta. Tell me, in SO2 will be there any lone pair? Or tell me, will be there any lone pair in CO2? Right? Please. So sulfur here is having how many valence electrons if I say? Again, sulfur 16th group element have 6 valence electrons. Fine. So here you will see sulfur. I am writing the 2 oxygen surrounding atoms. That's it. Oxygen make how many bonds? Very good. Yes. So sulfur, you will see, sulfur make how many bonds? Sorry, oxygen make how many bond? Double bond. Oxygen make how many bond? Double bond. We have to make bonds using the surrounding atoms. You should know the surrounding atom will make how many bond in general. Oxygen make double bond, so I made double here, I made double here. Out of 6 valence electrons, how many left? Out of 6, 4 are used in making bonds. 2 are remaining. So 2 are remaining, it become what? Lone pair. So yes, it is having one lone pair and 4 bonds it is having. Clear, bacha? Right? Very good. Rohit. In SO2 beta, there is one lone pair. Fine? Clear? Great. Now I am taking CO2. Now tell me the carbon is having how many valence electrons? 4. Carbon is having 4 valence electrons. Okay? So out of 4, first of all, you know 4 valence electrons are there. Now write carbon here, oxygen here, oxygen here. Now we know carbon want, we know oxygen want to make how many bonds? 2 bonds. Double bond, double bond. Carbon was having 4 valence electrons. Out of 4 valence electrons, all 4 are used in making the 4 bonds. Any lone pair will come here? No. There will not be any lone pair. Right? That's it. The simplest way I am saying. Right? Okay, we will see many examples like this for practice. If you know how to make structure, then definitely in the NEET exam, you will see 16 marks almost. 4 questions almost depend only on structure. Right? Maybe I will say, I am saying less only, maybe more than that. Because many questions directly, indirectly depend on structures. They can ask bond angle, they can ask bond order, they can ask bond length. All these things you can answer only when you know the structure, the VSC pair theory, which geometry will come. So everything depends when you know the center atom is making how many sigma bond, pi bond, or how many lone pair are present there. Fine? Clear, everyone? Okay, done. Next thing is, I am taking here, I am taking the ammonia. Okay, I think ammonia I took already, so I am taking H2. Right? And next you will see, we can write even OF2. Simple. Okay. If I think about the H2O, so center atom is oxygen. And oxygen have how many valence electrons? 6 valence electrons. Oxygen have 6 valence electrons. So please tell me, in H2O, yes, Rohit, Padma, Vishwanath, please tell me, in H2O we will have how many lone pairs? Go by this method. Right? You will see, in H2O what we have? O we are having here, H we are having here. So 6 valence electrons are there. And we know hydrogen will make how many bonds? Hydrogen to make how many bonds? 1 bond. We know hydrogen to make how many bond? 1 bond, right? So hydrogen making single bond, hydrogen making what here? Single bond. Out of 6 valence electrons, out of 6, 2 are used in making bonds. 2 are used in making bonds. How many remaining? Remaining are 4 only. So these 4 will be used as a lone pair. Getting my point? Sure. Next thing you will see OF2. Similarly, here also O is the center atom, 6 valence electrons, right? So here we have oxygen, fluorine here, right? So you will see fluorine, the surrounding atom, want to make how many bond? 1 bond and 1 bond. Getting my point? So oxygen used 2 of its valence electrons in making bonds, so 4 are remaining. 4 remaining means what? 4 remaining means 2 lone pair, that's it, finish. Getting my point? Yes, everyone? Okay, done. Now, remaining. So today we will do many examples for this part only. Now, the next example, if I say I am taking XCF2. Yes. Yes, Rohit, Vishwanath, Padma, Princess, tell me. In XCF2, how many lone pair are present? For your help, I am telling. In xenon, we have 8 valence electrons. Just write the xenon, write fluorine anywhere and just try to make bonds. Yes, everyone? Yes, tell me, in XCF2, we will have how many lone pairs? In XCF2, we will have how many lone pairs, right? Fast, fast. In HCF2, we are having how many lone pairs? Tell me. If you see, the xenon is having 8 valence electrons. Out of the 8 valence electrons, like first I'm writing the elements. Surrounding element, right? Surrounding element is what? Fluorine. Fluorine will make how many bonds? Fluorine will make single bond, we know that. Halogens as surrounding atom always make how many bond? One bond, right? So fluorine making single bond, making single bond. Out of 8 valence electrons, 2 are used in making bond for xenon. How many remaining? 6 are remaining. So 6 remaining become 3 lone pairs. That's it. Finish. Getting my point? Clear? Done. That's it. Next one. Okay. Next one I am saying. Now draw this one. I am giving you XCF4. XCF4. I am saying once again the step. You should know how many valence electrons center atom have. Center atom have 8 valence electrons. Put center atom here and then put the surrounding atom, and then you should know the surrounding atom will make how many bonds. In that way, you know the center atom used how many valence electrons already, and remaining valence electrons will make what? Lone pair. That's it. Clear? Yes, Rohit. Now, X I am talking about. X is having how many valence electrons? 8. And there are 4 fluorine atoms. I am writing 4 fluorine. You can write anywhere, no issue, because we are not discussing the geometry or shape here. I am writing 4 fluorines. I put the center atom. I know its valence electrons. It's very good. Now, next thing is fluorine will make how many bond? Fluorine makes single bond, single and what? Single. In that way, I know 4 valence electrons of xenon are used. So only 4 remaining. 4 valence electrons remaining means what? How many lone pairs? 2 lone pairs. That's it. So xenon is making here 4 bonds and 2 lone pairs, right? Clear? Yes. Please try same way. I am giving you next is XCO3 and I am giving next one is XCO4. So please. Yes, everyone tell me. Tell me, XCO3 is having any lone pair or XCO4 is having any lone pair? Please answer that way. Fast. Fast. Done. Very good. Okay, look at this. First of all, we are looking for XCO3. Fine. Xenon here, oxygen here, oxygen here, oxygen here, right? Tell me, oxygen, first of all, xenon have how many valence electrons? I am writing again. Xenon have 8 valence electrons, right? We know that. Now, oxygen want to make how many bonds? Whenever oxygen is present as surrounding atom, or normally also, oxygen make how many bonds? 2 bonds, right? So I am making 2 bonds here, 2 here, 2 here, 2 here. Out of 8 valence electrons, xenon used its 6 valence electrons in making bond. How many remaining? 2 remaining. So 2 become what? 1 lone pair, right? And in the end, we can check that xenon is an element of not element of second period. If it is not an element of second period, it can make more than 4 bonds, right? Only the second period, beryllium, boron, carbon, nitrogen, oxygen, these cannot make more than 4 bonds. Remaining element, I told you previously in third point, xenon can make many bonds, fine, right? Next one is which one? XCO4, fine. But what is XCO4? This is xenon here, right? I am writing oxygen, oxygen here, oxygen here and oxygen here in XCO4, right? So again, you know the oxygen want to make how many bonds? Oxygen want to make double bond, right? So oxygen is making here double bond here, double here and what? Double here. Getting my point? So in this way, if you see, oxygens make how many bonds here? Total 8 bonds. And xenon was having 8 valence electrons. So all the 8 valence electrons are used in making these 8 bonds. So any lone pair will left here? No. No lone pair will left, because all the 8 valence electrons are already used. Clear? Done? That's great. Now next thing I am taking here, we are having here CO3 2 negative. I am taking some ions this time. Okay, we are taking here CO3 2 negative. Fine. You will see, tell me, carbon. Carbon have how many valence electrons? 4 valence electrons. Carbon have 4 valence electrons. Fine. Right. So if I write here carbon as my center atom, and I will write here surrounding atoms, and how much charge is present? 2 negative. So definitely out of carbon and oxygen, oxygen is more electronegative. This negative charge is present where? On oxygen here, like negative and negative, right? 2 negative is present, one negative on one oxygen, the other negative on other, right? Normally a neutral oxygen want to make 2 bonds, because it is having 6 valence electrons and it want to make 2 bonds to complete the octet. But O negative is already having 7 valence electrons, so it will make only how many bonds? 1 bond, no 2 bonds, right? So O negative will make here what? Single bond. O negative makes single bond. Neutral oxygen will make what? Double bond. The neutral oxygen will make what here? Double bond, right? So in that way you see, now look at the carbon. Carbon was having 4 valence electrons, and out of the 4 valence electrons, all 4 are used, because it is making how many bonds? 4 bonds. So if 4 bonds are, if all the 4 valence electrons of carbon are used, then there is no valence electron remaining, so it will have no lone pair. Clear? So in that way we can see carbon making 4 bonds with the 3 oxygens here, right? Next part I am saying here, imagine we are having here ClO4 negative, ClO4 negative, right? Again, our center atom, which one? Center atom is chlorine. Chlorine is having how many valence electrons? 7 valence electrons. I am saying once again, what are the steps to draw structure? First, write the valence electrons of center atom, put the surrounding atom, and now you should know the surrounding atom will make how many bonds. And as I told you, it's a very common thing all the time: hydrogen, halogens or oxygen. These 3 things will be your surrounding atom. Oxygen make 2 bonds, halogens make 1 bond, hydrogen make 1 bond. That's it. Finish. Most of the question you can go this way. Okay, I am writing here Cl. There are 4 oxygens. I am showing here 4 oxygens, right? ClO4 minus. So one of the oxygen have what? Minus charge, right? So if this oxygen is negative charge, that's why this O negative will make which bond? Single bond. O negative will make single bond. Now this oxygen will make what here? Double bond. Neutral oxygen make what? Double bond, right? This oxygen make what? Double bond. This oxygen make what? Double bond. That's it. Finish. And whenever your center atom is making many bonds, so don't forget to check that it should not belong to second period. Okay? You see, the chlorine is belonging to third period, and that's why you can say it can make more bonds, no issue. Second period elements only have restriction, or you can say first period definitely hydrogen is always 1, helium will not make any bond. The second period element will have restriction, they can make maximum 4 bonds, right? Okay, fine. This is your structure. Easy? Very easy. Now the next one, if I say we are having here sulfate SO4 2 negative, we can draw SO4 2 negative, right? Yes, bacha, tell me, in SO4 2 negative, again, first step is to write the valence electrons of center atom. Okay, first step. Next step, first of all, write the center atom and show all the surrounding atoms. surrounding like this way. Put center atom and put surrounding atom like this. Now I know this is SO4 2 negative. So definitely oxygen is more electronegative. So this negative charge is present on what? Is present on oxygen. So I am giving two oxygens as negative charge. Okay, I have done this part. Now I know that O negative will make single bond because already it is having 7 valence electrons. Fine. So this O negative will make single bond, and this neutral oxygen make what? Double bond and will make double bond. As you can see, center atom is making many bonds, so we have to check once. Is it belonging to second period or not? No. So no issue. Sulfur can make many bonds, so no issue. It can make here like 6 bonds it is making, it's fine. Sulfur can make 6 bonds. Clear? Right? Will be there any lone pair on sulfur? Will be there any lone pair on sulfur? Yes, Rohit. Sulfur was having 6 valence electrons. Out of 6 valence electrons, if you check beta, it is making how many bonds? 6 bonds. Out of 6 valence electrons, sulfur made 6 bonds, and that's why there will not be any lone pair. Imagine I am having 6 valence electrons, and I used all my 6 valence electrons in making 6 bonds, right? So there is no valence electron remaining, so there will not be any lone pair. Getting my point? No lone pair in this case. Clear, bacha? Right? Clear? Okay, fine. Next I am taking 1-2 more examples. Next is, here we have NO2 negative, right? And next you can take, I am taking here ClF3. Okay, I think ClF3 is done already. Yes or no? Okay, fine. And next I will take one more. You can take XCF6. Fine, right? Now see carefully. Once again, go by the strategy. Nitrogen here is having how many valence electrons? Nitrogen have 5 valence electrons. Fine. Write nitrogen in center. Show the surrounding atom somewhere you can show, like oxygen here. NO2 negative. So oxygen is having which charge here? Negative charge here, right? And now you see the negative charge oxygen to make how many bonds? Single bond. Neutral oxygen make what? Double bond, right? Out of 5 valence electrons of nitrogen, it used its 3 electrons in making 3 bonds. Now you will see the 2 remaining become what? One of the lone pair. Fine, easy. Next thing is Cl. Chlorine is having 5 valence electrons. Okay, chlorine is having here 5 valence electrons, right? I am writing here this way. You will see we are having here fluorine. I am writing fluorine and fluorine. Fluorine as surrounding atom want to make how many bonds? 1 bond. It will also make 1 bond. Out of 5 valence electrons of chlorine, 3 are used in making bonds. If 3 are used in making bonds, how many remaining? 2 are remaining, right? Sorry, what I am saying here. Chlorine is having 7 valence electrons. Sorry for that. Chlorine is having 7 valence electrons. Out of 7 valence electrons, it used how many? 3 it used. Out of these 7, 3 are used. How many remaining here? 4 are remaining, right? So out of these 4, if you have 4 remaining, so 4 remaining will be what? 2 lone pairs. Fine, I am saying once again, chlorine have 7 valence electrons. Out of 7, 3 are used in making 3 bonds. So 4 are remaining, so 4 become what? 2 lone pair, right? Good, very good. Now the last question for today is XCF6. Here xenon, you can see, xenon is having how many valence electrons? 8 valence electrons belonging to 18th group, right? Now you will see, I am writing xenon here and I am writing fluorine randomly, right? So there are 6 fluorines. Every fluorine want to make how many bonds? 1 bond, right? So xenon used its 6 valence electrons in making 6 bonds with fluorines. how many remaining? Out of 8 valence electrons, 6 are used, 2 are remaining. 2 valence electron remaining means what? 1 lone pair. 1 lone pair. Right? Clear? So this way you can draw structures very easily. There are many structures, right? We will see some structures tomorrow as well, and then we will start the hybridization. Using structure, how we can find hybridization very easily. Just in 1 second, using structure you can find hybridization, and that part we will see tomorrow. Okay, I hope this will help you. Okay then, bye for today. Bye everyone.